Well, good afternoon, everybody. For those of you who I have not met, I'm Mike Clegg. I'm Dean of the school, and it's my job to welcome you to this Dean's Lecture. And we hold Dean's Lectures for special occasions, and primarily when somebody is either promoted or appointed to the rank of professor at our school. And yeah, I always say this at the beginning, is that I'm a cynical, jaded person, and tend not to be impressed with things. But being appointed to this faculty of our school is not easy. But to reach the rank of professor is especially impressive. And it's a decision that's really not made by our school. It's made by colleagues around the world, and everybody knows the process where we send out letters to people in the field and get back letters, and then they're weighed by the committee. So it's really a decision that's made by people from around the world. So and to reach this bar is impressive, and we're especially happy today to celebrate Xiaobin Wang's appointment here as professor. And we had, I guess, about 18 months ago, right after she came, an endowed chair installation, and that was really right at the beginning of when you came. And now we're going to see sometime after what's happened in the interim. And when Xiaobin came, she wasn't a stranger to the school. She's originally from China, got her medical degree at Beijing Medical University, and then she went to Tulane where she got an MPH, and then came here and got a Doctor of Science in Maternal and Child Health. And at the end of that long research training, she decided that she really needed to get the credentials to practice pediatrics and be a pediatrician. So she left us and then went to BU and trained in pediatrics. And then eventually ended up at Northwestern, where we recruited her from, where she headed an endowed center at the Children's Research Center. And when she came, and she was incredibly well funded, I think at that time had over 50 grants, and was leading this endowed center. And I said, Bob Blum and I, Bob who recruited her, and Bernie Geyer actually chaired the search committee, he's here, and was on the search committee and recommended her and remembered her as a student. When we had that endowed chairmanship event, one of the things that was remarkable to me was that she's been in a lot of institutions, and at every institution where she had been, somebody came and spoke, her mentor spoke, and spoke in incredibly glowing terms about her character, her scientific abilities, and her ability to collaborate across programs. So normally when you leave an institution, people don't do that. But she had really inspired love and respect in everybody that she had worked with. So she came here and we asked her, why, you're leaving a very good position. Why do you want to come to Hopkins? And she said, to collaborate, because I need colleagues. I want to take advantage of the colleagues that we have here at Hopkins. And you'll see as she talks about her work, she has done that incredibly well. So now she has over 150 peer-reviewed publications. As you'll hear, she's very interested in the early life origins of diseases. She has established 3 birth cohorts and has in different geographic areas, and has looked at a variety of important scientific questions. One is food allergies, which, as you know, is burgeoning in incidence in the U.S. and around the world, looking at what are the gene-environment interactions that lead to food allergies, and then looking at the in utero and preconception, post-conception factors that lead to chronic disease. She's received a number of honors. I'll just tell you about a few. And I won't talk much about her research because she's going to do that, but she's a member of the Institute on Medicine Committee on Understanding Premature Birth and Assuring Healthy Outcome. She's a member of the Genomic and Proteomic Network for Premature Birth Research Advisory Board at the National Institute of Child Health and Human Development. And she's been a member of the steering committee for the Consortium of Food Allergy Research at the National Institute of Allergy and Infectious Diseases. And of course, serves on many study sections. So we were delighted 2 years ago when we recruited her, and now that she's been here for 2 years, we're even more delighted that she's here. So Xiaobin Wang.
Good afternoon, everyone. Thank you so much. Thank you so much for coming. First of all, I'd like to thank Dean Clegg for giving me this tremendous honor and opportunity. I'd like to attribute this great honor to all of my teachers and mentors throughout my life. I must also attribute this great honor to all the colleagues and students I have worked with throughout my career. Without them, I wouldn't stand here today. My topic today is about early life origins of disease. I know that some of you are experts in the field, yet some are new to this field. So I'm gonna first talk about the clinical and the public health significance of pursuing these lines of research. Then I'll present some of our past and ongoing research. What I'm hoping is that these examples will help illustrate the research progress as well as challenges and opportunities we have had over the years and going forward. Finally, I will offer my perspectives on how to move the field forward. There are many reasons why we should pursue early life origins of a disease. Today, I'm gonna just highlight top 3 reasons. The first reason is that we need to develop more cost-effective strategies to prevent and control chronic diseases. In the US and globally, we face a growing burden of chronic diseases, escalating healthcare costs, and striking health disparities. Let's take a this slide. Globally, chronic disease, highlighted by purple color, among the leading causes of death. Just point a few. The #1 killer is ischemic heart disease, which has increased 35% over the past 10-year period. The #2 killer is stroke, with a 26% increase. The #9 killer is diabetes, but remarkably, you can see it's with a 93% increase over the 10-year period. In the US, the picture is similar. Chronic diseases, highlighted by yellow color, are among the leading causes of death. The #1 killer in the US is heart disease. #4 is stroke. #7 is diabetes. Not only the morbidity and the mortality of chronic disease is increasing, but these disorders are happening at a younger and younger age. As you know, chronic diseases, once clinically diagnosed, typically are not reversible and require lifelong medical treatment. The economic consequences of chronic diseases are astronomical because of the combined burden of healthcare costs and the lost economic productivity due to illness and premature death. In the US, as you know, the escalating healthcare cost is one of the most urgent national issues. It is now close to 20% of the GDP. The urgency and the challenges we face are further underscored by the 2013 Institute of Medicine report entitled U.S. Health in International Perspectives, Shorter Lives, Poor Health. Here, I just want to highlight one sentence from the report. The United States spends much more money on healthcare than any other country. Yet, Americans die sooner and experience more illness than residents in many other countries. Just give you a concrete number. The red bars represent the United States. The other green bars is for other developed country. As you can see, the life expectancy in the US is shorter than other developed countries. This is the case for both males and females. A big question is, in the US, what accounts for this paradoxical combination of high spending on healthcare with lower life expectancy and a relatively poor health status? While there's many reasons, here I'd like to point out one important reason relevant to early life origins of chronic disease. As you know, infant mortality is an important determinant of life expectancy. Let's look at the ranking of US infant mortality in the world. In the 60s, in terms of the lowest infant mortality rate, US was ranked 12th in the world. But in 2009, the US was ranked 27th. It is likely that the relatively high infant mortality is an important contributor to shorter life expectancy for the United States. The next question is, what contributes to the high infant mortality rate in the US? Let's look at the top 10 leading causes of neonatal deaths in the US. As you can see, the number one cause of neonatal deaths is the disorders related to short gestation and the lower birth weight. For whites, a quarter of all neonatal deaths were due to preterm low birth weight. For Black, 1/3 of all neonatal deaths were due to preterm low birth weight. In parallel with the high infant mortality rate in the United States, you can see that the preterm birth rate in the US is the highest among developed countries. Again, the red bar is for US, the blue bar is for the rest of the developed country. Even more concerning, the preterm birth rate in US Black is among the highest in the world. So this is pretty much in parallel with the top 10 highest preterm rates in the whole globe. Not only preterm is an important cause of neonatal deaths, preterm births may also contribute to a range of chronic diseases over the lifetime. The well-established associations include chronic lung disease, asthma, neurodevelopmental handicaps. Recent studies have also linked preterm births with increased risk of cardiovascular disease, hypertension, insulin resistance, type 2 diabetes, and also more recently with autism. From Barker's seminal work to more recent adverse childhood experience study, it is clear that early life experience could have a great influence on mental and physical health throughout the lifespan. However, the mechanisms underlying the early life origin of disease remains to be understood. It is generally agreed that all the chronic diseases we face today are complex, multifactorial disorders. Compared to the past, the field of early life origins of disease has entered a new era of promise due to the rapid advancement in biomedical research and biotechnology. We have an unprecedented opportunity to integrate multi-level and multi-dimensional data to better understand the causes and the underlying mechanisms of chronic diseases. For example, we are now equipped with increasing ability to measure biomarkers at multiple levels and at significantly lower cost. The field of epigenetics has drawn particular attention In Hopkins, we have world experts in epigenetics. Here, I just want to highlight some important features of the epigenome that are relevant to our understanding of early life origins of disease. The epigenome, which regulates gene expression, is largely established in utero. The epigenome can be altered throughout the lifespan, but it is particularly sensitive during in utero and early childhood period. Most exciting is that epigenetic alterations are potentially reversible, which raise the possibility that the epigenome could serve as the basis of novel intervention or therapeutic targets. A highly debated topic is whether epigenetic alterations induced by environmental chemicals or stressors can be inheritable without changing the DNA sequence. So far, the data were all derived from animal models. If it is true in human, it would lead to a huge paradigm shift. A recent article in Science touched upon this exciting yet controversial topic. In light of time, I will not get into detail, though it is a fascinating article to read. My past 20-year research effort has centered on two aspects. One is establish longitudinal cohorts and the biorepository. The second is to carry out collaborative research. One major challenge we faced is that in order to investigate early life factors and their tempo and dose response relationship with the later health outcomes, we need longitudinal cohorts. We also need biospecimens to elucidate underlying biological mechanism. Over the years, I have worked with hundreds of investigators across multiple institutions to establish large study cohorts and a biospecimen repository. Here, I have highlighted the 3 major cohorts we have established. The Boston birth cohort now consists of over 8,500 mother-infant pairs who are enrolled in Boston. It is a predominantly urban Black and Hispanic population. The Chinese twin cohort now consists of 2,000 pairs of identical and fraternal twins, which were enrolled in rural China. The Chicago family cohort consists of over 1,000 families, about 4,000 subjects, with biological parents and their children enrolled in Chicago. It is predominant Caucasian population. These 3 cohorts represent different geographic locations, ethnicities, and the socioeconomic background. They cover age groups from birth to adults to seniors. They include parents, children, and siblings. They allow us to study a broad range of pediatric and adult health outcomes. To date, this study cohort have supported 16 NIH-funded studies, as well as the training and the career development of numerous graduate students, postdoctoral fellows, junior faculties, and have generated over 100 publications. Within these 3 cohorts, we have examined a broad array of early life precursors of adult disease, including preterm birth, obesity, asthma, food allergy, and early life. So within the cohort, the disease we have examined including preterm birth, obesity, asthma, food allergy, early life precursor of adult diseases such as diabetes, metabolic syndrome, and cardiovascular disease. Today, I will highlight for you some research findings, specifically relevant to the causes and the consequences of preterm birth. In retrospect, my interest in low birth weight and preterm birth began 20 years ago when I was a student here. I came across this 1985 Institute of Medicine report entitled Preventing Low Birth Weight. I was very much intrigued by the enormous public health impact of low birth weight. So I'll just read one sentence that really struck me. Low birth weight is a major determinant of infant mortality in the United States. Infants weighing 2,500 grams or less are almost 40 times more likely to die during their first 4 weeks of life than normal birth weight. Low birth weight survivors are at increased risk of health problems, ranging from neurodevelopmental handicaps to lower respiratory tract conditions. So before I move on, I just would like to clarify a few terminology for those not familiar with this area. Low birth weight is defined as less than 2,500 grams or less. Low birth weight can be a result of preterm birth, fetal growth restrictions, or both. Preterm is defined as less than 37 weeks of gestation in the US. The majority of low birth weight is due to preterm birth. For this reason, the second IOM report actually changed the title to Preventing Preterm Birth. My doctoral work at Hopkins focused on birth weight, gestation, infant mortality, and racial disparity. Here, the 3 publications came from my doctoral work. My interest in low birth weight and preterm was further reinforced during my pediatric residency in Boston. The patients we served at the Boston Medical Center were primarily urban, low-income African Americans, a population with a very high rate of preterm birth at about 17%. My first pediatric residency was in the neonatal intensive care unit. Even though we have an increasing ability to save the very premature babies, I witnessed firsthand the tremendous impact of preterm birth on the babies and their families. These preterm babies spend their first postnatal life in the NICU rather than enjoying the love of their parents at home. If they did survive the first few months of life, too often they went on to suffer many long-term complications, including neurodevelopmental handicaps and chronic lung disease. It was clear to me that not only did I need to learn how to best care for the preterm babies, but also I needed to find ways to prevent preterm birth. A major obstacle we face in effectively preventing preterm birth is our incomplete understanding of the complex etiology of preterm birth. Inspired by the first IOM report on preventing low birth weight, I have been searching for the answers. 20 years later, I had the honor to serve as a committee member for the second IOM report entitled Causes, Consequences, and the Prevention of Low Birth Weight, Preterm Birth. It was very rewarding that our team's work has made some contribution to better understand the causes of preterm birth. Our team's work on preterm birth has contributed to the following aspect. It's including building the Boston birth cohort, gaining a better understanding on the role of genetics and the gene-environment interaction in preterm birth, exploring non-traditional risk factors of preterm birth, and investigating the linkage between preterm birth and the chronic diseases. In the following, I'll give you some examples. For those who are familiar with these research areas, 20 years ago, most preterm research focused on psychosocial environmental risk factors, the role of genetics and the gene-environment interaction in preterm birth were largely unexplored. Our team has conducted a series of studies to fill in this gap. Our work including familial aggregation study, candidate gene studies, African ancestry studies, gene-environmental interactions, and more recently, Genome-wide association study. So in the following, I'll give you some concrete examples. Our team was the first to demonstrate a strong familial aggregation of low birth weight and preterm in the United States, which may be attributable to shared environmental factors, genetic factors, or both. This work, published in New England, set the stage for our subsequent effort to establish the Boston birth cohort and incorporate genetic accessibility and gene-environment interactions in preterm research. Back to 15 years ago, large-scale GWAS was not feasible because of the high cost. Our team conducted the largest candidate gene study of preterm birth at that time. Our study provided some of the early evidence that the genetic variations may play a role in the risk of preterm birth. Our team addressed important questions at the time, that is, whether the adverse health effects of environmental exposures varies by maternal genetic susceptibility. For example, maternal smoking during pregnancy is a well-recognized risk factor of preterm birth. If, based on the conventional epi study, though, the odds ratio of preterm birth associated with maternal smoking was only about 2. Our team was the first to demonstrate that a subset of mothers with 2 variant genotype, C1P1A1 and GSTT1, if mother has a variance in these 2 genotypes, the odds ratio of preterm birth associated with smoking is over 10. So at that time, that was very striking. This is the highest odds ratio ever reported for preterm birth. Another area we addressed is in the US, Black women had the highest rates of preterm birth. Our team was the first to evaluate sequential predictive model of preterm birth by including conventional epi variables along with African ancestry and the gene smoking interactions. Our data showed that adding genetic markers and the gene smoking interaction improved the prediction of preterm birth. However, the improvement was only modest, indicating more works remain to be done. The candidate gene approach, as you know, is limited by what we already know about pathogenic pathways of preterm birth, which is still very incomplete. GWAS is now considered the most powerful approach for discovering novel susceptibility genes of complex human diseases. We are currently funded by NICHD to conduct one of the large-scale GWAS of preterm birth in African Americas. This is a preliminary plot from the first 1,000 subjects. Of course, this is underpowered. We're in the process of genotyping another 1,000 subjects. Even the type 2,000, we're going to face the same challenges as other GWAS studies have found. In other fields, for example, large-scale GWAS studies are now consistently suggesting highly polygenic models for complex diseases. In other words, such a model may involve thousands of genetic variants, each conferring only a modest risk, but collectively, they could be explaining substantial variation in disease risk within populations. Studies by us and others have also shown that the genetic and that a gene and environmental interaction may also play a very important role, indicating the importance to incorporate both gene and environment in understanding the causes of preterm birth, even in the case of a GWAS study. Therefore, the future GWAS study needs to go beyond the major gene effect model, and to incorporate both environment and genetic factors. Epigenome is believed to be at the critical interface between gene and environment. Evidence from animal models have implicated the intrauterine period as the most critical time for the establishment of epigenetic variability, which in turn influences risk for a range of disorders. Our work in the Boston birth cohort have demonstrated considerable individual variations in DNA methylation, which is detectable at birth and dynamic in the first 2 years of life. We also found that maternal obesity can alter offspring's DNA methylation in multiple genes that may have important implications for future chronic diseases. Let's back to the environment. We need to go beyond traditional risk factors. Why say that? For example, well-recognized risk factors for spontaneous preterm birth include Black maternal race, prior history of preterm birth, genital urinary tract infection, and maternal smoking, and a low socioeconomic status. However, studies have shown that these risk factors can only explain a fraction of spontaneous preterm birth. Another gap in the field is, in the past, studies tended to examine risk factors one or few at a time. What we need is to examine multiple risk factors simultaneously to understand their joint or interactive effect. For example, in this study, we showed that high psychosocial stress, this is the maternal perceived high psychosocial stress, and chronic hypertension can act in combination to increase the risk of preeclampsia up to 20-fold. Preeclampsia, as you know, is a common pregnancy complication and an important cause of preterm birth. We have also examined a range of environmental chemicals that are not obtained by vital records questionnaire, that cannot be obtained by vital records questionnaire or medical records. Our team was among the first to demonstrate adverse effect of air pollution on preterm and low birth weight. Our team also demonstrated a significant gene-benzene interaction on the length of gestation. We published a series of papers on the adverse effect of preconception serum DDT, DDE level on the spectrum of reproductive outcomes. Recently, we performed the first mother-infant paired studies that demonstrated remarkable maternal to fetal transplacental transfer of toxic metals, including lead, mercury, and selenium. And we also showed high degree of fetal bioaccumulation of mercury. We also examined various nutritional factors in relation to reproductive and pregnancy outcomes. Those nutrition factors include pre-pregnancy BMI, placental weight, preconception plasma folate, B6, B12, and preconception hemoglobin and ferritin. One interesting area recently has received great attention is whether nutrition and environmental chemicals can jointly affect health outcomes. However, most previous studies have tended to look at nutrition and environmental toxins separately. Yet women are often exposed to both, and the food intake may contain both. So as shown in this slide, we explored the joint effect of DDT and B vitamin levels in relation to early fetal loss. So we divided the women into high versus low DDT exposure and the folate level as adequate versus deficient. As you can see in this slide, the highest bar is the group of women who are exposed to a high level of DDT and also has a deficiency in folate. The bar next to this tallest bar, although this group of women also exposed to a similar high level of DDT, but because they have adequate folate status, their risk of early fetal loss was reduced. So our data provide the very first evidence that nutritional status may potentially alter the health effect of environmental toxins. So in summary, based on what we have learned so far, preterm birth is a complex multifactorial disorder. There's no single factor that can explain all of the preterm birth. Going forward, what we need is to simultaneously evaluate the role of the environment, genomics, and the epigenomic factors, as well as their interactions in preterm birth. What we are hoping is, in the process, we are also able to identify useful biomarkers and therapeutic targets so that we can better predict, prevent, and treat preterm birth. So now I'd like to switch gear a little bit and to talk about the link of preterm birth with chronic disease later in life. I'm going to use our very recent studies published in JAMA as an example. Most previous studies on fetal origins of adult disease were conducted in people born full term yet with low birth weight. In contrast, much less is known about the link between preterm birth and adult diseases. I think one of the reason for this case is because most previous study was based on the older people. Those people tend to born 60, 80 years ago. At that time, most of the preterm baby cannot survive. But today is a very different picture. Most preterm baby can survive. Therefore, the population burden of preterm births are increasing. For this study, our intent was to fill in an important data gap during the early developmental period, specifically the in utero and early childhood period. We used the prospective birth cohort design that is enriched by a spectrum of preterm births. We aimed to test the hypothesis that preterm birth is associated with elevated plasma insulin levels at birth and in early childhood. Our study included over 1,300 children from the Boston birth cohort who were enrolled at birth and followed prospectively from birth onward. As you can see in this figure, there is incredible inverse relationships between gestational age and the plasma insulin level at birth. The more severe the prematurity, the higher the level of blood insulin. This pattern persists into early childhood. As further demonstrated by this slide, children ranked in the top insulin category at birth were more likely to remain in the top category in early childhood relative to children ranked in the lowest category. Our study provides consistent evidence that preterm birth and perhaps even early term birth as well may be a risk factor for the future development of type 2 diabetes. What is the clinical and the public health implications of our study findings? So here I just would like to share a quote from the JAMA editorial in the same issue as our study. Studies such as that by Wang et al. Reveal just how early the first steps towards prevention of diabetes may be possible, and raise the prospect that rigorous studies of early life interventions could form an important aspect of helping to reduce non-communicable chronic disease risk. Before I conclude my presentation today, I'd like to offer some personal perspectives on pursuing early life origin of disease in general. We have made great strides over the past 20 years, yet there are still huge gaps to fill in. To move the field forward, we need a life course approach and longitudinal cohort studies. We need to leverage cutting-edge science and technology and integrate multi-level data. We need a strong interdisciplinary collaboration to bridge age groups and disciplines and bridge pediatrics and adult medicine. We need a shared vision among government agencies, academic institutions, and the general public about the allocation of resources and investment for the future. There are growing public interest as well as concerns. Our biggest challenges and opportunities will be how to translate the new scientific discoveries into clinical and public health practice, including early prediction, preemptive intervention, and individualized medicine. I'd like to emphasize that the complex etiology of chronic disease and the clinical and the public health translation will require close collaborations among many disciplines across the country and across the globe. Only by working together can we realize the unprecedented opportunities that lie in front of us. I would also like to stress that the future belongs to the younger generations. Today's students will be tomorrow's leaders. We need to do everything we can to create a nurturing yet challenging interdisciplinary environment that allow junior investigators to grow and to lead. In summary, there is growing evidence on the importance of early life experiences on later health and diseases. The mechanisms underlying early life origins are complex, likely involving psychosocial, environmental, genetic, and epigenetic factors. Longitudinal birth cohort and a biorepository are necessary to understand the natural history, root causes, and the biological mechanisms of non-communicable diseases. Advancement in this field may help improve health for all ages and transform future clinical and public health practice and policy. Last but not the least, I would like to acknowledge grant support from the NIH and the various foundations. I would also like to thank those who have given generous philanthropy support, in particularly the Krieger Endowment and the Ludwig Family Foundation. I am indebted to all the institutions and individuals who have supported, and contributed to our research over the past 20 years. And finally, thank you all for your time and attention. I'd be happy to take any question you may have. Thank you.
So let me turn this on. So it's being recorded, so we should use the microphones for questions.
Oh, okay.
I'm sure lots of people have questions. Let me ask the first one. So when you look at low birth weight, you showed a lot of factors, that conceptual model.
Mm-hmm.
Have you tried an analytic approach where you put multiple variables, either in a sort of a hierarchical model or multi-level model, to see where the pathways are and what total risk is explained by variables in those domains?
Yes, we did. One of the papers I cited here, and we're trying to do that. We actually included 3 group variables, that including the traditional epidemiological variable, that include various maternal prenatal variables, and we also included African ancestry and the gene smoking interaction. So we're trying to go beyond the traditional risk factors. And so based on the ROC curve, yes, there's an incremental, but the ROC curve is not great. That really means we still hasn't yet to identify all the possible risk factors. So this is the work we need to continue.
Great, thank you. So Stan, could you use the mic, please?
This is probably a naive question. So could you explain why is the insulin high in the premature infants, and what is the genetic link? Thank you.
So Stan, can you see what you want to understand?
Why is the insulin higher in the premature infants?
Why premature infants, it's more
Yeah.
in U.S. than other countries?
No. Why is it higher in the preemies? You showed the.
So the insulin levels are
Insulin
higher.
levels.
Oh,
with
I see.
premature.
Oh, you talk about insulin.
And what is genetic link for that? Thank you.
Oh,
Sorry, I
that's,
spoke too
wow,
fast. Maybe.
that's a great question. So step 1, what we found is we found association. So that's what our JAMA report is about, is association. So you're asking what is the mechanism? So I'm going to actually, I know here we have a great bunch of scientists. I was hoping to asking some of you help me. So we found association. I think the next step is what is the mechanism? Actually we looked in, but we couldn't quite figure it out, to be honest. So I need help.
Somebody want to take? She's looking at you guys.
So I'm looking at peers and.
But as you said, the insulin
Yes.
levels you're using them as a marker of insulin resistance.
Right.
So there's peripheral tissue resistance to the action of insulin in those children, right? So that's like the next step is they reflect that you need more insulin to have the metabolic effects of insulin
Yes.
take place.
It's remarkable what we demonstrated is even at the time of the birth, those preterm baby manifest insulin resistance. So we just said, actually after even adjusting for birth weight, adjusting for body fat. So the fact is there. The question is, what is the mechanism? So we look at the fetal growth. The pancreas actually formed in the first trimester. Is amazing how early that pancreas was formed. Then by second trimester, they're starting secreting insulin. But that insulin, who knows whether it's functional or not. Then there's that negative feedback loop. It's remember the largest weight gain happens third trimester. That linked to leptin. So what happens? Think about what happened to a premature baby. Most baby born in third trimester. So the third trimester baby, they are low birth weight. The reason they're low birth weight is they didn't have the chance to grow during the third trimester. And without growing, they don't have enough body fat. They're not enough leptin. So that kind of a negative feedback loop is not there. So we put this speculation in the JAMA paper, then the editor said, take it out. Because that's totally speculation, not proof. So we have to cut back from the mechanism explanation. I said, okay, I'm going to leave it to my colleague to solve.
Thanks, Xiaobin. I think a lot of us are really in admiration of so much of your work. I'm in particular admiration of your ability to start work with the Chinese in clinical settings. Could you tell us what the challenges and opportunities are for collaborating for research in China?
Excellent question. I have collaborated with China over the past 20 years. I feel, for me, it's primarily a very rewarding process. I guess in part is I'm very lucky we have wonderful collaborators. And together, we have investigated 3 areas. The first area is reproductive health. Second area is obesity, metabolic syndrome. And the third area is stroke. So in terms of reproductive area, we made a unique contribution because China has that unique one-child policy and also family planning policy. So that means all the preconception women, if they wanted to have a baby, they have to report to the official and register before they can conceive. So because that a unique policy and opportunity, we are able to study preconception factors and how preconception nutrition, preconception environmental toxin could affect early reproductive outcomes. As a result, actually, one of the I'm showing, that's what we have. And secondly, in terms of reproductive health, is we studied air pollution. Everyone knows how bad the air pollution is in China. So we were among the first to demonstrate. In fact, at that time when we published our study in environmental health perspective, we said we found that the linkage between air pollution and the low birth weight and preterm birth. Someone think, what's that? We never heard about it. And so what's the mechanism? So yes, we didn't quite figure out a mechanism. We didn't make that observation. And subsequently, this became a very hot topic. In fact, I looked at my citation of that paper. It's like a 400 or 500. It's one of my most highly cited publication. So that's an example of unique opportunities. And in terms of obesity metabolic syndrome, what happened is in China, the lifestyle changes, and their lifestyle towards more sedentary, more Western. There's more obesity and more metabolic syndrome, diabetes. And so we worked together to establish the twin cohort, and that allow us, number one, to study the longitudinal changes and also use a unique twin design to tease out the gene and the environment influence on the obesity. And the third area is the stroke. The stroke, as you know, actually in China has the world's highest rate of stroke. So it's number 1. In China, stroke is the number 1 cause of death. US is number 4, and in the whole world is number 2. The China, why have such a high rate of stroke is one is because there are so many people have hypertension. Secondly is Chinese people have an incredibly high homocysteine. And this high homocysteine in hypertension patient is about 70–80%. The reason is so different from US is because the 3 reasons: low intake of folate and a high mutation of MTHFR gene is the C677T mutation. So that mutation leads to high prevalence. That's one of the reasons. So we're actually, I participated with one of the largest clinical trials ever in the history about primary prevention of stroke in China, involved 20,000 subjects. We're able to demonstrate, it's not published yet, but I think the preliminary data has already shown that folate supplementation in the country without folate fortification can significantly reduce the risk of stroke. So overall, my experience with China is when we're collaborating with internationally, number 1, we need to focus on what's the major clinical public health issue. Secondly, it's leverage complementary resources and unique population characteristics that create unique opportunities and will not exist in either US or China, but combined together, we can do some wonderful things. And I know many of you engage in international collaboration, and certainly you have a lot of wonderful experience to share as well.
So it's running a little late. Maybe we'll do Bernie and then end with Bob.
So, Xiaobin, I was going to ask you, and maybe others in the audience would also say something about what's the impact of this kind of work on cardiovascular epidemiology as it's being conceived of by most of the people who work in that field. So we know that early on, the Barker ideas were pretty much discredited and controversial and didn't have a lot of impact. But you know, the body of kind of evidence that you're talking about goes so far beyond the origins of this early origin work that I wonder, is it beginning to influence, you know, the usual work on risk factors that people, you know, smoking, diet, cholesterol, what kind of impact is it having on that work?
Bernie, I just have one disclosure. Bernie was my thesis advisor. We have some conflict interest. Okay. So Bernie raised the great questions. If we think about, sorry, I need to be there. If we think about how the history of medicine progressed over the years, it's starting from symptomatic treatment to disease treatment to the future vision of preemptive, preventive, personalized medicine. So this is like a progressive vision and the progressive improvement of the practice. And if we think about how science has improved over years, think about from, take genetic as example, 60 years ago, the DNA double helix was discovered, but it takes about 50 years to finally get to the point a human genome was sequenced and cataloged. And for the Barker hypothesis, in fact, I looked at history. Barker wasn't the first person had this idea. It's back to 1930s. And what happened? First person raised this idea, fetal origin, but didn't quite take off. And the Barker hypothesis actually was named by BMJ in 1995. That means 60 years later, Barker finally was able to gain the public recognition and receive more attention about this work. Use his data. I think evidence probably is also important. I guess early people, maybe they don't have the solid scientific evidence. Barker was the one not only continued to advocate for this idea, but he used the data. And his data was very compelling. But I think the fact, even if you look the Time magazine, and the public start to get excited. And I think maybe at the Barker time, if there's multiple Time magazine about that, maybe he were getting more popular at that time. So what I'm saying is the science is a process. It takes ceaseless effort. And if it's the truth, it will be a truth. And keep pursuing it. Don't give up.
Story and then a question for you. So connecting Bernie with Xiaobin, when Bernie retired, in this very room, we held a FET trip, and there were some extraordinary people who came and spoke. There was a student of his who had also been invited to speak, but she had been injured and wasn't able to fly from Chicago here to honor Bernie in person. So she decided that what she was going to do was present her talk live by speakerphone with the slides being projected. So all we heard was a voice and the slides. Is that not true? Xiaobin gave this astounding presentation, astounding. And at the end of it, I walked up to Bernie, I said, who was that person? Who is she? And might she ever consider coming here? So we're pleased you did. My question, going back to the JAMA article and the preterm, the relationship of preterm to insulin levels. And you said that that continues at least into early childhood. So one thing I'd be interested is, what do we know about that trajectory? Secondly, what do we know at this point in time between the relationships epidemiologically between preterm birth and obesity over time, and between biophysiologically the relationship of that insulin trajectory and obesity? Because obviously insulin dysregulation and obesity are sort of related.
Okay, Bob always gives me challenging work to do. Yes, what we found is, so let me explain what we did, what we hasn't done, what need to be done. What we did is fill in a gap. And two New England Journal paper, what they did is they did a retrospective study. See, they were able to link type 2 diabetes, insulin resistance to retrospectively asking people, were you preterm? So they made that connection. Even with that kind of connection, they made into New England Journal paper. And so we look at that data, we said, wait a minute, there's a gap there, because what happened at birth and what happened in the first, in the early childhood, there's no data. So what we did is we tried to fill in that data gap. So that's what we did. We demonstrated is, okay, if you are born preterm and the insulin level is higher at birth, and also this higher trajectories persisted into early childhood. What we didn't prove, which we're hoping we were able to prove in the future, is if we continue follow this cohort into adolescents and adults. In that way, we're able to prove with more data point. So by that time, I think we can provide definite answer. If it's not named the Barker hypothesis, we're gonna hopefully name the Barker plus hypothesis or something. So that's about preterm birth. And to talk about the obesity issue, actually it's very interesting. So actually we are currently working on it, hopefully another important paper. What we found is people would ask me, okay, if I'm born preterm, I'm doomed to be diabetes, can I do something? And is there any way we can change the fate? Yes, my answer is yes, if we work on it as early as possible. So one area we potentially can work on it is to regulate how much baby should gain weight during the early period. So currently we examine the weight gain postnatally, first 4 months, the first 6 months, 12 months, and 2 years. And what we found that surprisingly, people hasn't looked this very carefully, is the very first few months of life weight gain actually has a very important predictability for the future development of obesity. If you look at the New England Journal recently, there's recent New England Journal paper on childhood obesity. What they are able to demonstrate is there's a linkage between kindergarten obesity and the adolescent obesity. That's their finding. So what we demonstrated is, we're also trying to fill a gap here, is what happened to the first few years of life. What we are able to demonstrate is, if a child, regardless your preterm low birth weight or SGA or LGA, as long as you gain excessive weight, you're going to be at extremely high risk to become obese at the kindergarten. And also the kindergarten obesity will persist into elementary school. So this is another work we're currently working and wrapping up, and I hope we can share with the scientific community.
Thank you. So on your last slide of acknowledgments, you listed the Ludwig Foundation, Family Foundation. I want to recognize Carol Ludwig, who's in the room here, and for your talk. And so thank you for your support. And in the Q&A, what comes out to me is your intense passion and your intense logic. And so we can carry those two things out to where the reception is, and people can enjoy that. So thank you.
I want to thank you all, truly. I know how busy you are, and that the weather is not so good. Thank you so much.